Oh, konbanwa. It is time for me to stop procrastinating and finally study again. I don't want to do this, but I have to. I don't have a choice. So, today I'm studying neurological anatomy, which is going to be the choice of my pain for today. I have to watch like 7 lectures in like 4 days, and they are like 3 hours long, so I need to watch on a pace of 2 lectures a day. Um, yeah, I'm not in a good spot. So, anyways, I'm gonna be watching for like 6 hours probably. We'll see if I make it alive. I don't know if I'm gonna really live through this, but we'll see. We'll see how far we can go. I'm only predicting that I'll probably finish 1 or 2 lectures. If I'm lucky, maybe 1.5. We'll see. Anyways, that's gonna be what I'm up to. I hope everyone has a good rest of their day. Ooh.
The nature of that sensory stimulus. So we start with the periphery, then we move to the cranial nerves. Of course, we spend quite a bit of time on the cranial nerves and the processing of information from the face, sensory information from the face, and then motor as well. So with that, the exam review is right after class today, so definitely encourage you to come to the exam review. Overall, I think the exam went very well, I've been 83. So again, if you feel like you're struggling, please
Central
just reach
nervous
out
system?
to me.
Yes, it is.
Okay, so with that,
Mm-hmm.
we'll go ahead and dive
Spinal
in
cords
to
and
the
all that.
nervous system. And this image is an image that you saw, I think, on the very first day of class when I was talking about the nervous system. But just to kind of remind you, and now you're so much further, right? You've done all the neurophysiology, but at any rate, thinking about what are we talking about for the rest of the semester. We're spending most of our time talking about the central nervous system. So
What
when
the
we're
heck is
talking about the central nervous system, it's the brain, which
PDHPE?
includes the brainstem, and then the spinal cord. We're talking mainly about the central nervous system, but we will generally talk about, as I mentioned, we'll talk about pain and temperature to start out. Certainly that sensory information is traveling from the periphery into the central nervous system. So just
Oh my
putting together
goodness.
what we've talked about up to this
Yeah,
point with Dr.
it's
Cassina, when you were
so
talking about
painful.
skin, you talked about nociceptors, those free nerve endings. You talked about Purkinje corpuscles and Meissner's corpuscles. So at any rate, out in the
Excuse me.
periphery, whether it's out in the face and you're touching your face and that information is carried by a cranial nerve or one of these spinal nerves, right? The peripheral nervous system is going to communicate with the central nervous system.
By the end of this four
Everything that
days,
you see
I will
here,
know
just a
everything
couple, those are the
about
details we're going to cover
literally the
in
nervous
anatomical
system.
sciences too, up in the gross anatomy lab
It's
next
gonna be
semester.
crazy, chat, I
But at any
promise.
rate, when we're today, we're going to really talk about
I'm
just
gonna
the
go
structures
crazy.
of the central nervous system. But just keep in mind, as we talk about these various pathways, we're going to
Or I'm gonna
start
be
with
dead.
sensory
It's one of the
pathways,
two.
taking information into the central nervous system and how it's processed. Again, how we know that, maybe I burned my finger versus dropped something on my toe, right? So we'll start with the sensory pathways, and then we'll move on to the motor pathways. When we think about sensory pathways, we have somatosensory, so that's like light touch and pressure and pain and temperature. We have special sensory, that would be vision and taste, and we'll talk about all those sensory modalities. And then visceral sensory information. If you have a stomach ache, that's visceral sensory information, or information that passes, from the periphery back to your central nervous system, that allows for changes out in the periphery. Again, secretion from glands, for example, or temperature information to impact a motor, like sweating, for example. And then for the motor system, we've talked about the voluntary motor system. We talked about skeletal muscle, so we refer to that as somatic motor. And then we have autonomic motor. Okay, so that would be associated with smooth muscles and glands. Okay, so that's an overview of just kind of thinking about the peripheral nervous system and the central nervous system. And again, they interact. That interaction between the central and the peripheral nervous system is critically important. This is a slide. This is actually the last slide as well, and I put it as a first slide just to give you a sense of an overview of the central nervous
Oh
system.
my
and what we're
god,
talking
there's so
about today.
much.
So when we think about the
Ah.
central nervous system, I already mentioned that we have the brain and the spinal cord. We'll talk about the brain and the spinal cord today. But what you can see is there are further subdivisions. These are general subdivisions. So we'll talk about the brain stem. So the brain stem includes 3 levels, and you can see them here in green, but we'll talk about the midbrain pons and the medulla quite a bit. I think in every lecture we'll mention some part of the brain stem. We also have the cerebellum. We don't say much about the cerebellum until the second half of the course. We have a whole lecture on the cerebellum, but this structure numbered 1 back and through here is the cerebellum. And then we have the cerebrum. We have 2 cerebral hemispheres. So here we're looking at this image. This is as if the brain is cut
That's the
straight down the middle.
cross-section.
We'll look at many sections like this. Here we're looking
I
at a
don't
brain
see
that's
where the
cut,
cerebrum is.
in what we call the coronal or the frontal plane, giving us front and back pieces. But at any rate, what we can see is that we have these 2 cerebral hemispheres. So we can see with each cerebral hemisphere, we have either 4 or 5 lobes. You can see this textbook talks about a limbic lobe, but we have a frontal lobe, parietal lobe, occipital lobe, and temporal lobe. We'll talk about all of those. We have another structure deep in the brain, or a group of structures deep in the brain. I mean, you can see the term here, basal ganglia. So the basal ganglia, we have a whole lecture on the basal ganglia, but these are nuclei, a group of nuclei deep in the brain. I'll show you some of the basal ganglia a little bit later this morning. But when we think about that terminology, right, we use the term ganglion to represent a collection of cell bodies or to describe a collection of cell bodies outside the central nervous system, within the peripheral nervous system, right? A ganglion. Here we have basal ganglia, which is a collection of nuclei in the central nervous system. So it's a bit unfortunate, but the name actually comes from a developmental term.
Is location.
It develops from the
Ganglia
eminence.
is a
So at
bunch
any rate,
of
we'll talk about,
neural
and I'll show you a
cells.
few of these nuclei, but we're going to focus mainly on the basal ganglia later on in
It's
the
a
course.
group
And then we have some
of
other structures, the
neurons
hippocampus and a
in the
medulla,
brain.
which we will talk about much later in the course. So
So
at any rate, we have the
backside
cerebral hemispheres, and then we have the
neurons.
diencephalon. The diencephalon includes the thalamus and the hypothalamus, are the 2 main structures. They're the only 2 structures I'll mention today, but as we move through the course, we'll also talk about some other structures of the diencephalon.
Oh, great.
So at any rate, what
Lovely.
we can see generally here is just this sort of breakdown of all the different regions, starting from the central nervous system and sort of making our way deeper into the brain and more specific. Okay, so I think that's just sort of a nice general overview of that image from your textbook. So what are we going to talk about? Well, we're going to sort of march through the central nervous system today. We're going to start with terminology. So we're going to start with some terminology. Some we've used, some we haven't. But when we think about the central nervous system,
Hey, my boy is here.
Lecture, we're going to be talking
Guess who's
about
rewatching?
really
Guess who's rewatching the base level lectures all over again?
Okay.
This guy.
So
I still don't get it.
You're going to come back to
And guess who watches at freaking 1.0? This guy.
Are familiar to you. We probably have said them.
I still don't get it, dude.
Occasionally during the course as well.
I have to like rewatch the lectures like every like 3 days
What you can see
just to understand it.
Superior is always
I wish you can see this right now, my man.
Is always toward the feet. It's like it's always superior is always up, sort of the most towards the head or towards the cranium, and inferior always going to be sort of lower down towards the feet. And it doesn't matter if it's in the gross anatomy lab,
classmates.
the spinal cord, the brain stem, or you're looking at the cerebral hemispheres. When we talk about the terms anterior and posterior, anterior is always toward the front. So your abdomen is anterior to your backbones, right? Posterior is always nearest the back. So again, it doesn't matter where in the body you're talking about. that's just how it's organized. And it's always organized based on anatomical position, which is something like this, right? Just standing. Okay. There are other terms, however, where body region is important. That's okay. Body region is important. So dorsal and ventral. So when we are talking about just gross anatomy next semester, when we're looking at the spinal cord and we're looking at the brainstem, ventral is toward the front and dorsal is toward the back, right? So here, what you can see is that ventral is synonymous with anterior and dorsal is synonymous with posterior. That's if we're talking, in this course, really the spinal cord or the brainstem. But what we're going to see when we get to the brain itself is that now dorsal is synonymous with superior and ventral is synonymous with inferior. So why is that? I should have brought a prop, right? Piece of paper here. All right. So if I didn't come prepared today. Okay. So think about fish, right? So let's say these red X's, right? Again, over the course of evolution, right, we have like fish and, maybe a squirrel, mouse, right? And you think about like a dorsal fin, right? A dorsal fin, right? So the fish is like this. So the spinal cord, right, and the top of the brain, they're in a straight line, right? So think about a dorsal fin. This is what you don't want to see when you're swimming on Cape Cod, right? So that dorsal fin, the top of the brain, is continuous with the back. But in humans, what happens? We can see as we look at this particular image that as the brain develops, we get this bend in the brain. So fish were like this, where the X's represent the back, and the ventral surface, where there are no X's.
It's Zip. Hello,
Where there are no
welcome.
X's, it kind of, right? Where there are no X's here, that's the ventral surface. But in humans, what happens? We're upright, and then there's a bend to the brain, right? So what does that mean? That what used to be on the back is on the top, right? So now, for the spinal cord and the brainstem, the back of the spinal cord and brainstem are dorsal, but the top of the brain is the dorsal surface of the brain. And think about, I think for dental students it can be helpful because you have talked about have you talked about the dorsal surface of the tongue?
Top of the brain is dorsal.
And actually the top of the foot is the dorsum of the foot. So I think about those things that stick forward. I mean, our brain kind of projects forward.
Got it.
And our tongue projects forward and the top of our foot projects forward. That's the dorsal surface. So you can see that this is the dorsal surface, and then all of a sudden the top of the brain is the dorsal surface. And that means the undersurface of the brain is the ventral surface. So just think about this sort of being a hinge here. Okay, so we've got where we are is really important. So you can see here when we're talking about dorsal and ventral, body region is important, right? So dorsal is the top, and the ventral surface is the undersurface, just like the undersurface of the tongue is the ventral surface. Okay. Rostral and caudal, those are not terms that we use in gross anatomy, but we use them all the time in neuroanatomy. So when we're talking about the spinal cord and brainstem, rostral is synonymous with superior and caudal is synonymous with inferior. So what does rostral mean? Rostral technically means toward the beak. We don't have beaks, so you can think about it as toward the nose, right? So you,
Towards
if
the
my
beak.
brainstem, right, or your brainstem, or this brainstem here is rostral to the spinal cord because it's above, it's closer to the nose.
Rostral.
We don't have tails. Caudal technically refers to toward the tail, but toward the feet you can think about.
Caudal.
Okay. So for the spinal cord and brainstem, rostral synonymous with superior, caudal synonymous with inferior. Okay. Now, right, when we get to the brain, now the beak is, right, the nose is going to be synonymous, rostral is going to be synonymous with anterior, right? So this part of the brain here, which happens to be the frontal lobe, is anterior or rostral to this area back here, which is caudal or posterior. So when we get to the cerebrum, rostral and caudal, right, that position, because of the bend in the brain, rostral is synonymous with anterior, caudal posterior. So I think this is a good time to, I was going to ask if there are any questions, but see if
Is it a good time to take a break?
identify the dorsal surface of the brain. So it should be Whitney, should be and I did put this slide in, right, because it's so important. I don't want to wait until the exam. So I did put the image in. So like on your
Top
own
surface
image,
is
you can mark
dorsal.
the correct answer. Okay. So when we looked at this previous image here, brain. So rostral toward the beak.
Okay.
Perfect to the nose.
I need
So
to
when we look
get
at
a
this
blanket. I'm actually
previous
freezing
image
up in here, boys.
This, oops, this polka dot is rostral or anterior. These back and through here are
My
caudal
poor
or
friend.
posterior.
It's so
the
funny.
top of the brain
He texted me
is
and
the
he was
dorsal
like, I've been watching lectures all
Any
day,
questions about
and I came by to check your
any
stream,
questions about that?
only to see that I'm watching more lectures.
You know, mental gymnastics to sort of
It feels
think
so
about,
bad.
the directions. Okay. So
How poor
planes
thing
of
had to
section,
witness that.
again, we have, I know we've thrown some of these words out, but we're going to say them a lot. These images on the right, I saw online. I thought, oh, that's really clever to sort of think about maybe if there's a ways to remember the various planes. Some of you, I know you're very comfortable with this, but the sagittal plane, so the idea was that Sagittarius has a bow and arrow, but at any rate, when we think about the sagittal plane, right, it gives us right and left pieces. I also like to think about what's left, like when you cut it. So if you cut something in the sagittal plane, you're left with a right piece and a left piece. We'll see that there are times when we're looking at the brain, we have equal right and left pieces. And in that case, it's a mid-sagittal section. What we say when it's off of midline is a parasagittal section. So we will occasionally look at some parasagittal sections. The horizontal plane, another name for the horizontal plane is the transverse plane. But when you think about a horizon, right, horizontal, it's at any rate this flat surface that would cut something, giving a top piece and a bottom piece. So this is in the horizontal plane here. So we would have a top piece and a bottom piece. And then coronal. So think of crown, although there are other terms like the frontal plane, so frontal and coronal. That gives us a front piece and a back piece.
What's the frontal?
So those terms, or these terms noted on the slide, we're going to see these over and over again, various sections of the brain. So you'll get comfortable with those various sections. The other term that we'll use is cross-section. So for the brain and the spinal cord, often people just talk about a cross section. That's just by convention. And that's in the horizontal plane, giving us, again, a top piece and a bottom piece. So today we'll look at some brainstem cross sections and spinal cord cross sections, and it's just right across those regions. Okay, so this brain has been cut in the...
That's frontal. It's the frontal plane. Is it not frontal?
Nice.
Am I
Okay.
dumb?
So all of the top 5, right? So coronal, coronal plane, frontal, frontal coronal, coronal frontal. All of those are correct. So
no, wait, I'm not
giving us
dumb.
a front and a back piece is the
It's just
coronal
people
or
kept on calling it
frontal plane.
coronal.
Those are synonyms. Parasagittal, that would give us right and left pieces. So parasagittal,
Oh my god.
this one is not correct. Parasagittal would give us, would be sort of a cut this way that would give us right and left pieces. Any questions about that? Okay. Just a couple other terms that you probably know, but we're going to use them over and over again. And ipsilateral, contralateral, bilateral, and unilateral. Okay. Ipsilateral means that something's on the same side. So we can see here in this example, the red dot and the green dot are ipsilateral to one another. They're on the same side. If something's contralateral, now what we can see is that the red dot and the green dot, right, they're on opposite sides, so they're contralateral from one another. And that's going to become important when we talk about various pathways. We're going to say that a pathway can cross to the opposite or the contralateral side. So we're going to see that quite a bit in neuroanatomy. Most of the pathways that we talk about are bilateral, right? Just like I have pain coming in from my right hand, I have a different pathway coming in from my left hand. So the pathways might cross to the contralateral side, but I have 2 of them. I have one coming in from the right side of the body and one coming in from the left side of the body. So as we move through the course, almost everything is bilateral and symmetric, but occasionally there's something that's only on one side, right? So that would be unilateral. And so again, we'll get there, but these terms will come up over and over. Oops, okay. All right, so we're done with terminology. And now we'll move on to the cerebral hemisphere. So we're going to talk about the cerebral hemispheres, and you'll use this term telencephalon a bit more in your development lecture in January, but telencephalon also refers to the cerebral hemispheres, and you're going to see that there's a telencephalon during development as well. Okay. So let's start off by talking about the gross structure of the brain, what we can see with the naked eye, and then we'll get deeper into the brain. Okay, so here we're looking at a lateral view of the brain. So here we can see, this is a view just from the gross anatomy lab here. We will take the brains out of the cranial cavity in gross anatomy, and we'll study the brain next semester in your hands, but here we're just looking at it this semester at various
To touch
images.
it with my hands.
So
Gross.
what we can see here, this is the anterior aspect of the brain. Here's the posterior aspect of the brain. When the cerebellum is intact here, as we can see, it's quite easy to tell what's the anterior part of the brain and what's the posterior part of the brain because the cerebellum is posterior. Okay. So at any rate, in this image, we can see the cerebrum, which is all of this is the cerebrum. We can see the brainstem, and then posterior to the brainstem is the cerebellum. So they're showing the cerebellum here in this image, and the brainstem. And from this lateral view, you can't fully appreciate that we have these 2 cerebral hemispheres. And I did make this is just from the Society for Neuroscience website, but right, you can just see, I just sort of took this little video, so it'll twist. And what you can see is that there are two cerebral hemispheres. And what you can see is that like you could put your hand between the two cerebral hemispheres.
Oh, that's so
but
dope.
you see that there's a structure. I'm going to stop that. Right, there's a structure deep in through here, and we'll take a look at that very shortly. That's a communication between the right and left cerebral hemispheres. But again, we have these two cerebral hemispheres, and then it's just going to turn the opposite direction. Okay, so really the point is that we have two cerebral hemispheres. Gross anatomically, they are symmetric. Again, there are some functions that are lateralized to one side, but the brain in many respects are quite, the two parts of the brain are symmetric in terms of processing sensory information and then sort of motor output. But of course, there are differences that we'll talk about later in the course. Okay. So here we're looking in this upper image, we can see a lateral view of the brain. So here we're looking at a lateral or a side view of the brain. And here we are looking at a medial view of the brain. So this lower image, it's as if we cut right through the brain. And what we can see is that there's this region right here that we cut through. And in fact, this, do you have this image? I feel like,
no, we do not have, we don't.
Okay, I can post this, but this is a mid-sagittal section. So this is a mid-sagittal section, and this is where you would put your hand. I'll post this, but this is where you would put your hand, right? And you would go down, right? You put your hand between the two cerebral hemispheres, and then you would bump into this structure right here. Okay? So this is that structure I was mentioning that communicates between the two cerebral hemispheres. And this structure is the corpus callosum. So it is shown, I did note it on this slide here, the corpus callosum. So this is the corpus callosum, which here is shown in yellow. Okay? So that corpus callosum, right, is that communication bundle between the right and left cerebral hemispheres. Okay? So what you can see in this image and in this previous image is that you're not cutting through cortical tissue here when you're cutting the brain. It's not until you get to the corpus callosum. But then what you can see is that in order to get a view like this, you had to cut through the brainstem and you had to cut through the cerebellum. Right? You can actually see that there's like a slice through it. And that they are trying to depict that in this image as well, where you can see they cut through the corpus callosum, and then this is the brainstem and the cerebellum. Okay. So as we look at the brain, we can see that it is not smooth, right? We have the evaginations are gyri, and the invaginations are sulci. So gyrus and sulcus, those are singular. The terms I just used, gyri and sulci, are plural. And this has occurred over the course of evolution. And you do have this next slide when we look at the brain of a rat compared to
that's my
human.
brain right there.
So, you know, the human brain has 86 billion neurons, so we need a lot of surface area. So
smooth.
Over the course
like
of development, there's a point in development where the brain is smooth and then it expands. And so we have, just like we have like microvilli, what does it do? It increases surface area. We have all of these sulci and gyri, so we can walk around with 86 billion neurons in a reasonably sized skull, right? Otherwise, we'd have a big giant skull. All right, so we have these folds, the sulci and gyri. We'll look at some of the names. You'll see the term fissure. Sometimes we use the term fissure. Those tend to be particularly deep sulci. Okay, so we have 4 lobes here shown in blue. We have the frontal lobe in this reddish color, the parietal lobe. You can see the frontal lobe and the parietal lobe both laterally, right, on this lateral view. as well as on this medial view, right? So here's the parietal lobe. The occipital lobe is the posterior aspect of the brain, and then sort of this inferior region is the temporal lobe, which we can also see on the medial surface. So those are the 4 lobes of the brain, and they are demarcated by various sulci. So the central sulcus, we'll be talking about the central sulcus on Tuesday, so we'll spend a lot of time up in this region of the brain in the next week or 2. But at any rate, we have the central sulcus that separates the frontal lobe from the parietal lobe. Then we have this sulcus, the lateral sulcus, also known as the Sylvian fissure, that separates the temporal lobe from the frontal and parietal lobes. So if I were to ask you, the temporal lobe is blank to the frontal lobe. So it is inferior. So it is inferior, and it's also
Ventral?
someone else may have said it, I just didn't hear it.
Is it
It's okay. So in terms of it being there, right now we're looking at a lateral view of the brain, so we can see this is actually
Ventral.
a lateral view. So everything that we're looking at is on the lateral surface. It's almost as if, just to orient you, like you took like my skull off here. This is exactly what you would be looking at, the lateral surface of the brain. So I probably should have written that here. This is a lateral view. What other term? So think about your tongue.
It's ventral.
It's ventral. It's ventral. This is ventral, right? Because it's on the sort of the inferior and ventral are synonyms here. Caudal would be back and through here. So caudal would be the back. Okay. All right. So,
Hi, Kyo.
okay.
Have fun studying.
So
I
at any
won't.
rate, we looked at the
I absolutely
lateral
will
sulcus, we
not.
looked at the
But
central
hi,
sulcus, and
Yue.
then as we
Thanks for
looked
stopping
medially,
by. I hope you've
we
had.
can see that
I hope
there's
you've had a good day. Thanks for hanging
sulcus
out and saying
between
hello.
the parietal lobe and the occipital lobe, appropriately named the parietal occipital sulcus. We don't actually have a really obvious demarcation laterally between the occipital lobe and the parietal and temporal lobes. Okay, so, and then just the last fissure, the longitudinal fissure, that's what I showed you in this previous image. This is the longitudinal fissure. So the longitudinal fissure just separates the right and left cerebral hemispheres. Okay. So just one more question. This is my last direction question.
Ventral.
This is kind of a hard one because we just started, 15 minutes ago.
15 minutes ago? I feel like we've started like 5 billion years ago. What the fuck?
Okay, let's see how you did. 100%. Excellent. Yes,
Holy
100% of you got it right.
amazing.
Yes, it's the ventral surface. It's the undersurface of the brain. And this is hard because I haven't even showed you the undersurface of the brain, but it's really the surface we haven't seen yet. Yeah, so here we're looking at, the front part of the brain, right? Here's the brainstem, and then back and through here, this is the cerebellum. Okay, good. All right. So that's the gross structure of the brain. Now let's take a look at the white matter and the gray matter. So we have talked about white matter and gray matter. We started there back over 2 months ago now, right? 2.5 months ago when we were talking about the nervous system, histology. Okay, so what are we looking at here? We're looking at, so this is a lateral view, and I probably should have labeled it here, but this is a lateral view of the brain, which makes this anterior and this posterior back and through here, or this rostral and the occipital lobe back and through here is caudal. And what I'm trying to show you is that, right, the section below is a coronal section or frontal section. And this is what it looks like. This is what the brain looks like with the frontal or coronal section. So here's that lateral fissure. Again, we have those two cerebral hemispheres. And then here's that bundle of axons that's communicating between the two cerebral hemispheres. So this bundle, right, this is that bundle, this is that corpus callosum that I showed you previously. But really what I wanted to show you here is the organization of the brain and what is referred to as the cerebral cortex. So you know at this point in
I don't know how to spell
your
corpus
studies that
callosum.
cortex refers to bark. That's the word origin. So we talked about the cortex of many organs up to this
Callosum.
point. So the cortex, right, the cortex of the brain is just this outer surface of the brain, right? And that is gray matter. So someone looking at a fresh specimen of the brain thought that looked gray, and they called it gray matter. Right? This is before we knew what was there, which is neurons, and then what's in through here is white matter.
Oh, for the love of God.
So when I say this is gray matter, right, does that mean just what I've outlined right here? Is this A, neurons, like cell body? I'll just put CB for the cell body of a neuron. Or is it B, the axon of the neurons? It is the cell body, yes, the cell body. So gray matter refers to the cell body. The white matter refers to where we have the axons. And I think back in August, I mentioned the way that I remembered it. I mentioned like white, think white matter is fat. It's the myelin sheath, which is the phospholipid bilayer. And so, right, it's the wrapping of the axons, the myelin. And so it looks white, just like if you're vegan, I apologize, but even if you're just looking at a piece of bacon, how do you know where the fat is? It's white. If you're looking at a piece of steak, how do you know? It's white, right? So think about that. So white matter, axons. Okay, so the cortex is where we have the gray matter. All right. You'll have a whole lecture on the cortex later in the course, but just to introduce you to what is in the cortex, in the gray matter, well, we have layers of neurons. We have most of the human cortex, I have that here, most of the human cortex is a 6-layer cortex. Right? So, and we call that 6-layer cortex the neocortex. Neo refers to, I sort of think of it as like newer over the course of evolution. We have some brain regions, not that many, that are 3, 4, or 5 layers, but most of the human cerebral cortex has 6 layers of neurons. Okay, so the neuronal cell bodies, as I mentioned, are on the outside. That's the cerebral cortex. And then we've got lots of white matter deep in the brain. But we do have some groups of nuclei, right? So when you see these sort of brown areas here, this means that there are cell bodies here. And so some of these are nuclei of the basal ganglia. Some of these are thalamic nuclei. We'll talk about those as we go through the course. But the cortex refers to the outside of the brain where we have all those neurons. And here we're just looking at an image of the brain and the 6 layers that we see in most regions. So you can see that, here you can see layer 1. This is a Nissl stain, right? So this is a Nissl stain. So you can see we don't have that many neurons. Layers 2 and 3 actually look quite similar. If you sort of look at the types of neurons, there are some differences we won't talk about today. But then you can see there's a distinct layer 4, there's a distinct layer 5, and there's a distinct layer 6. So we see these layers to the human cerebral cortex. and kind of interestingly, this is just an aside, like most of the human cortex is on average about 4 millimeters, which when you think about just the processing power of the human brain, it's just like the outer 4 millimeters or so. And it does vary between just about 2.5 to 4.5 or so, but it's not really that thick. And you can appreciate that in this previous image as well. Okay. So
Oh my God.
over the course, so for many hundreds of years, people have had this idea that there were functions associated with the human brain. And people started to kind of map these different areas of the brain. And this is a historical drawing of sort of these various regions. And this was this mapping was what's sort of referred to as a pseudoscience. It's not a real science. It was sort of like a made up. And people cross ethical boundaries as they develop these maps. So essentially what they would do is they would feel the skull and say, you know, these are the different functions. So I think it's interesting because you can see this idea of like esteem and cautiousness and courage. But also people made suggestions about biological differences between groups when clearly no biological differences exist. And so it's sort of looked now, sort of looking back in history, as like a very sort of terrible way that sort of research went, as I have here. It's like it wasn't real research. Again, the idea was that they were just feeling bumps on the skull. And that's in contrast to Brodmann, who was a real scientist. And so that's really what I want to mention is that Brodmann, so back in the early 1900s, very methodically looked at sections of the brain and was able to identify subtle differences in these cortical areas. So, for example, maybe in one area, he noticed, sorry about that, he noticed like particularly large neurons here, right? And that was in this sort of special motor region. And then some other regions, he noted these very subtle differences. And he developed this map, which has been come to known as Brodmann's map or Brodmann's areas. And the interesting thing about his map is that it has, well, stood the test of time right now, over 100 years. And the reason this map has stood the test of time is because his identification of these subtle anatomical differences, what we call like cytoarchitectural differences, like how the cells are organized, correlate to the physiology. So when he noticed, well, oh, this area has like slightly bigger cells, I'm going to number it this number, it turns out that that's a specific functional area. So in this course, we will talk about Brodmann's areas, and I will show you this map over and over again. As future dentists, I don't ask you to memorize the numbers. So I might mention, you know, I don't know, primary visual cortex is area 17 on an exam, and it might be written, you know, on a slide. I would put both of those in there. Again, as dentists, I don't think you need to know the numbers, but I do think it's important to understand they exist, because if you look in the literature, the neuroscience literature, neuroscientists use it all the time. If you're, I don't know, reading about a patient with a stroke or something like that, you might come across these numbers. Okay, so those are the various areas. And now I want to talk a little bit about some of the demarcations of the white matter. So here you can see, does anyone know what kind of section this is? Yes, it's a sagittal section, and then specifically, it's a parasagittal section. And again, so that would probably be a good thing to write down. It's a parasagittal section, and the way you know it's parasagittal, it looks like you can see that we've cut through that cortical tissue. So when I showed you
Get it.
this image previously, you can see, right, there's there wasn't a cut made through the cortex itself. Okay.
I see.
So this section allows us to see deep in the brain and to see that white matter. Okay. So here's the gray matter along the didn't need to do that, along the outside. And then what we can see are these axons that are just either streaming down from the cortex to deeper structures, or axons that are streaming up toward the cortex. And just a couple of terms. There's what's called, sorry about that, the corona radiata, which someone thought that this region looked like a radiating crown. So it sort of looks like that, right? Like a crown would look. But the structure that's really important is this internal capsule. Okay? We're going to talk about the internal capsule quite a bit, because what we see is that axons from a very broad cortical region get packed together and they pass through this region called the internal capsule. And I'll show it to you on the next slide as well. And it's particularly important because what you can imagine, if, let's say, the axons in here, just between these two red, or just where I colored this red in, a stroke in this area, right, could be similar to a stroke in this very wide area, right? Because these axons are coming down and they're getting packed together. So a small stroke in the internal capsule can have devastating clinical consequences because they just all come together in this small area. So we will talk about the internal capsule. And what we can see is that there are many parts to the internal capsule. I'm going to ask you to be aware of 3 of them right now. So what we're looking at, let me just orient you to this image. What we're looking at is essentially a horizontal section through the brain. So this would be anterior or rostral, right? Because this would be up and through here, right? This would be toward the nose, right? This was like a person. So this is rostral. And we know that in this image, because we can see the cerebellum here, at any rate, we can see that there's some deep gray matter. These are actually nuclei of the basal ganglia that I mentioned previously. So we can see what we're looking at here are the axons passing from the cortex. Now, this is a myelin stain. So we know it's a myelin stain because the white matter, the axons are a black color. So at any rate, this stain imparts a black color to the axons, and essentially the axons are coming down through the cortex, and then they pass through this region, the internal capsule. And when I look at the internal capsule on a view like this, it sort of looks like a boomerang, right, or an invert, like a V. And so we have what's called the anterior limb, which is where the A is. We have the posterior limb, which is where the P is. And genu refers to knee. So when you think about your knee, think about bending. So at a bend, this here is the genu, right at the bend. Okay, so this is the genu right here. And then we've got the anterior limb, which is more anterior, and the posterior limb, which is more posterior. So we won't worry about the other parts of the internal capsule, but that's the internal capsule. It's like the white matter, right? The axons are coming down from the cortex, and they come together, and they pass through sort of between these nuclei that are deep in the brain, and we call it the internal capsule. Okay. So just one thing. Like I said, we are taking a tour through the brain today. Almost everything every structure that I'm talking about, we are going to come back to. So next week, when we're talking about pain and temperature, I'm going to talk about the internal capsule, right? So we'll be talking about like various regions of the brainstem that we'll move on to. So if all of these words are new to you, I get that anatomy, there's a lot of new words. Just go back and review it and know that we are going to come back to all of these regions. So when I talk about pain and temperature, I want you to have at least heard of the different sections of the brainstem, and I want you to have heard of the internal capsule. So we're going to apply the pathways to these various regions. Okay, why don't you take a 10-minute break? We'll get back together at 10:32. The regions within. I want to talk a little bit about the ventricles because we are going to see the ventricles in almost every section that we look at. Those are the regions within the brain that have cerebral spinal fluid within them. So they're going to look like big spaces in the brain. So you've already seen some of the ventricles, and I just want to point them out at this point. Later on in the course, we have a lecture, and we'll spend a fair amount of time talking about the ventricles and talking about the production of cerebral spinal fluid. But now I just want to give you a general overview, again, so you just know what they are as we're making our way through these various sections. So here we're looking at a mid-sagittal section. This is out of the Netter Atlas that we use quite a bit in gross anatomy. And so this is that corpus callosum that I mentioned previously, but when you look at this image, you can see that there are spaces here within the, sort of deep within the cerebral hemisphere. This space here, well, it's the lateral ventricle, and you can see I put a 2 next to it because there are 2 of them. There's one associated with each cerebral hemisphere, right? So these are the spaces. Many of you have heard of cerebral spinal fluid. I'm going to assume that all of you, because you've probably talked about it, but at any rate, we have cerebral spinal fluid that's produced here in the lateral ventricles. Okay, so what are we looking at here? Here we are looking at what's called the single midline third ventricle. And I'm assuming that we have, like someone thought, okay, well this is 1 and this is 2, and so this is the third ventricle, right? So we don't have a first ventricle or a second ventricle, we just have 2 lateral ventricles. Okay, so that's, I think, where the third came from, right? But we have this third ventricle. The third ventricle is a single midline space. Where is it located? Well, it's located medial to the thalamus. So I mentioned the thalamus previously, and you've talked about the thalamus in physio. So here's the thalamus, and so here you can see the bulk of the thalamus on both sides, and there's this space between the thalamus, and it actually extends here between the hypothalamus. So this is the hypothalamus, another structure you talked about in physiology, but this is the third ventricle in through here. So as we're looking at this mid-sagittal section, you can see I'm pointing the space here. This is the third ventricle. It would be coming out of the board because the thalamus would be deep in, right? So we're looking at just this medial surface. Is everyone with me that when we're looking at this mid-sagittal section, let me just erase everything. When we're looking at this mid-sagittal section, we're just looking at this surface of the thalamus. It's as if you're standing and you're looking at the thalamus this way. So this third ventricle, right, the space of the third ventricle is out of the, not tall enough, but it's out of the screen here. Okay. So we can actually see it quite a bit better here in this coronal section because it actually looks like a space here. Any questions about that? Yeah. Oh yeah, so here, these are the lateral ventricles here and here. So they're associated with the cerebral hemispheres. They're deep in the cerebral hemispheres. And then the third ventricle, so you can see it's not between the cerebral hemispheres. We're coming down to this region I'll talk about later, the diencephalon. So I kind of introduced you to that on that first flow diagram, but the thalamus is part of the diencephalon. So you can see we're kind of making a step down. And then what we can see here in this mid-sagittal section, we can see that from the third ventricle, which would be where I have this red dot, we have a continuation, right, where cerebral spinal fluid can now flow through this cerebral aqueduct, which we can see passes through a portion. This is a portion of the brain stem. And then it passes into what's called the fourth ventricle. Right? So we have two lateral ventricles. We have a third ventricle, which we can, where I just put the 3. Then we have the cerebral aqueduct. And I think I'll use sort of a yellow here to represent the cerebral aqueduct is right here. And then we come to this larger space again, which is called the fourth ventricle. So we'll look at these over and over again. Just again, at this point, I just want you to understand that we have these ventricles. They're filled with cerebral spinal fluid. In a lot of images, we're going to see these spaces. Okay. The last point I want to make is that the fourth ventricle is continuous with the central canal of the spinal cord. So you can see that yellow line on the slide, right? You can see that there's a sort of a thin line that's representing the central canal of the spinal cord. This does occlude after birth. In most adults, it's fully occluded, but at any rate, central canal of the spinal cord. How cerebral spinal fluid then flows from the brain out, we're going to worry about later on in the course. Okay. So we'll look at a few more images today, and I'll point out the various spaces associated with the ventricular system. Okay, so we'll go ahead and move on to the diencephalon. So I use that term diencephalon a few times. So the diencephalon, so we have it, when we talk about the cerebral hemispheres, I use the term telencephalon. Again, a developmental term. Diencephalon is a different region that includes the thalamus, which you can see here with the T. This is the thalamus. This is a similar image to what I showed you previously on the previous slide. And this is the hypothalamus. So these are two of the structures of the diencephalon. There are other structures we'll talk about later. But what we can see is that they're deeper. And just one point I want to make, and you can see I noted it on the right side of the slide here. Oops. That we cannot see the diencephalon with an intact brain. With an intact brain, we cannot see the diencephalon. This is what we see, right? This is a lateral view. This would be anterior here, right? So this is anterior, but this is a lateral view. The other side would look exactly the same. We cannot see the diencephalon. In order to see the diencephalon, we need to section the brain in some way. So here in this upper image, you can see a mid-sagittal section. And then in the middle panel, right, the middle panel, we've just sort of zoomed in on this region that includes the thalamus and the hypothalamus. So again, here you can see the thalamus. This is that corpus callosum I showed you previously, the thalamus, and then the hypothalamus, hypo below, so below the thalamus. Okay. So as we were previously looking at the ventricular system, right, I mentioned that, right, this here, this space here, where I just put this red, what space is this? The third ventricle, exactly. So this is the third ventricle. So you can see the thalamus and the hypothalamus. So at this point in the course, right, so you want to have a sense of where the thalamus and the hypothalamus are. They are part of the diencephalon, and we see that they border the third ventricle. Okay. The thalamus and the thalamus and the hypothalamus, they're both made up of a group of nuclei. And that you have a sense, I think, from physiology. You've talked about a few different nuclei associated with the thalamus. We will also talk about some nuclei associated with the thalamus. At this point, what you can see, I just want to go through this image on the left. So here, this is the cerebellum, which is sort of the back of the brain. What we're looking at here, a portion of the brain stem. And so you can see, they're showing you here in green, this is the thalamus. And what they're trying to illustrate for us is that there are different nuclei of the thalamus. You will learn some of these nuclei. I think you've talked about a couple of them. We'll talk about a few more. Again, you're responsible for the nuclei as we move through the content. But for our purposes now, just having an understanding that we have different nuclei. I remember thinking that the thalamus was just like this homogeneous thalamus, and then to learn that there's like lots of different nuclei, I just remember being very surprised. But at any rate, we have this organization of these various nuclei, and then this information heads up to the brain. So the point that I want to make here is that as we talk about sensory pathways, we're going to see almost every sensory pathway has to go to the thalamus before it gets to the cortex. So sometimes it's referred to as the gateway to the cortex because it has to sort of make a stop. There's a processing within the thalamus, and then it heads up to the cortex. And this particular image, this is from the textbook. This is just illustrating for us by color coding the various organizations. So this region here that's shown in green goes up to this region of the brain. There's another lighter green region that goes up to this region of the brain. This orange nucleus goes up to this region of the brain. So we'll talk a little bit about some of the organization, not every single nucleus. Okay, but having a sense of, again, where the thalamus is, it's part of the diencephalon, borders the third ventricle, there's an organization to it, and it's the gateway to the cortex. That's really the take home for the thalamus right now. In this course, we don't talk much about the hypothalamus. It will come up a couple of times in a few lectures, but you've talked about the hypothalamus and its importance in homeostasis. So at any rate, really the only point that I want to make is that like the thalamus, the hypothalamus is also comprised of many different groups of nuclei. Each nucleus has different function, and so I think we'll just leave it at that. Okay. All right. So we started at the brain, right, the telencephalon. We went to the diencephalon. Now we're moving down to the brainstem itself. Okay. Oh, here's that image. All right. So you do have that image. So here we're looking at a nice image of the brainstem. So again, previously, this is probably the best image that I showed you, though, of the corpus callosum. Right? So that's a nice image of the corpus callosum. And we can see this would be the thalamus, right? And this would be the hypothalamus, right? So these are the structures of that diencephalon. What you can see here, this is just a minor point, but we don't see into the lateral ventricle because there is this really fold of connective tissue between them, okay, the right and left lateral ventricles. And we could see that, I think it was, yeah, so you can see that. It's called the septum pellucidum. It's not a term you're going to be responsible for, but you can see that they actually are separate. So that's why we can't look into the lateral ventricle here. There's just a fold of tissue there. Okay. But what I want to introduce you to actually on this slide are the three regions of the brainstem. So what do we see here? Well, we see this upper portion of the brainstem called the midbrain. As we make our way inferiorly or caudally, we can see that there's a portion of the brainstem that has sort of this bulbous ventral portion. And this is the pons. So all of this here is the pons. And then where it tapers again, we have the medulla. And then the medulla is continuous with the spinal cord. Okay, so we're looking at the three parts. Oops, I didn't mean to do that. We're looking at the three parts of the brainstem there. We can also see, this might be a good place to label the ventricles as well, right? So this would be the third ventricle. What you can see here is that when I mentioned the cerebral aqueduct, I didn't say this previously because I hadn't introduced you to the midbrain, but the cerebral aqueduct goes right through. See that? It goes right through the midbrain. So again, this is the third ventricle up and through here. Then we have this narrowing, right? Cerebral spinal fluid passes through this narrow region. That's the cerebral aqueduct. And then the ventricular system opens up again into this fourth ventricle. So as we look at various cross sections, we're going to see that the cerebral aqueduct goes right through the midbrain, and then behind the pons, right, posterior to the pons, is where we see the fourth ventricle. So we're going to see the fourth ventricle between the pons and the cerebellum. So we'll actually see that in a couple of images. Okay. So just one other point that I'm going to make. I'm going to go ahead and erase this. That I mentioned that the cerebral aqueduct goes right through the midbrain, which means that this tissue back and through here is midbrain, and this tissue here is midbrain. So there's just like a hole that goes through it. And so when we look at the midbrain, which we're going to do in just a moment, what you're going to see as we look at some sections, you're going to see these little bumps off the back, right? So I'm exaggerating them, but you'll see these little bumps off the back. And these are, I don't think you've talked about them. Have you talked about, no, maybe you did talk about the colliculi. Did you talk about the colliculus, the superior colliculus with the visual system? Does that sound familiar? Okay, well, here's your old friend, the superior colliculus. That's it, right there. All right. So I'll show you an image of that in just a moment. Again, we're going to talk about the superior colliculus. We're going to talk about these. I'm just introducing you, trying to orient you to various brainstem cross sections. So what you can see, so in this mid-sagittal section, we have a really nice view of the brainstem here. Right? On a whole brain, we don't have such a great view, especially of the midbrain. So what we can see, these are the temporal lobes. Right? So I mentioned the temporal lobes kind of peak. This is what we refer to as the medial part of the temporal lobe. But at any rate, the temporal lobes, they kind of cover up the midbrain. But this is the midbrain. Then you can see this bulbous portion of the pons, and then down and through here we have the medulla. Okay, so this image is a very close-up image looking at the brainstem and the various cranial nerves. Now you can see on this slide that I'm not going, some of you, and you did talk about some of the cranial nerves, I'm not going to ask you to just memorize the list. We are going to talk about every single cranial nerve every single cranial nerve except 11 in neuroanatomy, and every single cranial nerve next semester as well as we talk about gross anatomy. By April, these are just going to roll off your tongue if they're not already rolling off your tongue, because we're going to be talking about cranial nerves so much. So there's no sense in just asking you to memorize a list right now. We will talk about them as we move through the course. But really the point that I do want to make as we talk about the brainstem is that cranial nerves 3 through 12 emerge from the brainstem. Okay? So cranial nerves 1 and 2 are either associated with the telencephalon or the diencephalon, and then the cranial nerves are just numbered from rostral to caudal. So, if we're just to look at them, right, so we've got 2 and 3 and 4 and 5. So you can see I'm pretty much just making my way from rostral to caudal, 6, 7, 8, 9, 10, 11, and 12. But really the point I just want to make right now is not having you just memorize a list out of context, but just understanding that these are where the cranial nerves are emerging. Just like I had showed you previously, the spinal nerves emerging from the spinal cord, we'll talk about that, but these are just nerves that are going into the periphery, right? So carrying, pain from a tooth, right? You burn your tongue, right? Or the roof of your mouth with some pizza, right? That's carried by a cranial nerve. A spinal nerve is carried by carrying information from my hand if I touch a hot stove. Okay. Any questions? Yeah. Yeah, so cranial nerve 1 they're really associated with, The cranial nerve 2 is associated with the diencephalon, so that's the optic nerve. So you probably talked a little bit about that in the neurophysiology. Cranial nerve 1 is an exception. It's really associated with the telencephalon, but it's one of those that it's actually in the nasal cavity and just goes into the optic, the olfactory bulb. So it's associated with the telencephalon, but it's actually in the nasal cavity. Yeah, it's kind of interesting. We will talk about it. It is one of the, and the interesting thing about, this is a total side, don't write this down, but when you take the brain out of the cranial cavity, you always tear the olfactory nerves because you're taking the brain out and they actually go into the nasal cavity. So there's no way to
Nixie,
take out the brain without
hello.
damaging
Welcome.
the nerves
How are
themselves.
you? How have you been? Thanks for stopping
So
by.
when we talk about the olfactory bulb and the olfactory tract, which you might have talked about somewhere along the way, those are not the nerves that are actually carrying
How's
the
your
information
weekend
from
been?
the nasal cavity. Okay.
I'm studying right now for
we are now
anatomy.
going to look at cross sections of the brainstem. We're going to look
You've
at cross sections of the brainstem
been...
in every
Yeah. That sounds about
lecture
right. That sounds
for
about
the
right.
next at least many
Well,
weeks, at least until the next
if you're
visit.
wondering how I am,
Okay.
I mean, do you see what I'm
The
having to do
brainstem
right now?
cross sections. The brainstem cross sections that I show you right now are the same brainstem
This is
cross
my
sections that
life.
you
This
will
is a
see on
sneak
the
peek of what
exam,
my life
which
is on a
not
day-to-day
always
basis.
the case with, image of the spleen or something like
Absolute
that. These will
fantastic.
be the same ones. They are all myelin stain. The myelin stain is a purple stain in these brainstem cross sections. They are right from your neuroanatomy textbook, the Nolte neuroanatomy textbook.
Is it a requirement for schooling?
A
Yeah,
little bit
it's
ago,
what's
I
it called?
showed you a myelin stain where the
It's
myelin
dental
was
school.
black. It was just a different kind of stain that imparted
So
a black
it's a
color
little
to the
bit
axon.
of
All
a
of the
sneak
brainstem
peek
cross
of
sections
what my
that I
life
show
is.
you
I have an exam
impart
for
a purple color to the axons. So
neurological
we have
anatomy.
previously looked at the Nissl stain that imparts a purple color, so you want to know that these are myelin stained sections. I am not showing you any Nissl stained brainstem cross sections. They are all myelin stained. When I first looked at brainstem cross sections, I saw a lot of purple and a lot of white. And
Good luck. Thank
there's
you.
a lot
Thank
of
you.
purple
Thank
and a
you.
lot of white. Okay. But each section, it is worth spending some time
We're learning
and
about
thinking
brains
about
and
each
shit.
section. What is the feature that allows
Thanks
you
for
to
lurking. I
identify
appreciate
this
it.
as a particular section?
Have a good rest of your day.
So this upper image is the rostral midbrain. So, you can see here I have a mid-sagittal section. So this is a mid-sagittal section. Again, it would probably be worth just writing down mid-sagittal here. So this is a mid-sagittal section through the brainstem here. Okay? And what we can see, right? Well, the first thing that I notice is that there's a hole. Okay? So that takes us back to the point that I previously made, and that is that the cerebral aqueduct, right? So this is the cerebral aqueduct. It goes right through the midbrain. Behind the midbrain, we actually have, that's where we have these bumps. These are the colliculi. So this happens to be the superior colliculus. You're not responsible for that name yet, but you can see these prominent regions posteriorly. So at any rate, the midbrain is the only location where you're going to have a hole, and that's that cerebral aqueduct. The other structure that jumps out at me, maybe even the most, are these. These are called, again, you're not responsible for the name quite yet. These are called cerebral peduncles. Someone thought they looked like little feet. Don't they look like little feet sticking out? Can you imagine, like little feet sticking out? So those are called cerebral peduncles. So like feet of the cerebrum, I guess. Okay. So at any rate, these are cerebral peduncles. And find something, whatever it is, find something The three things I just pointed out, we're not going to see at any other level. So when we look at the pons, what do we see at the pons that's unique? Well, the pons has this very bulbous ventral region. So that it's fairly easy to identify. The other feature is that behind the pons, we have this big space, and this is the fourth ventricle. And what you're actually starting to see right here, I might have another image where I have more of the cerebellum. This is actually the cerebellum back in through here. I mean, you don't want to identify it by the cerebellum, but you can see you go through pons, fourth ventricle, and then cerebellum. But this bulbous portion of the pons, we're just not going to see that morphology anywhere else. Okay. For our purposes, for the exam, you are responsible for the rostral midbrain. It looks like I did put a caudal midbrain in your image deck if you want to take a look at it. But these are the four that are on this slide. These are the ones that I'm going to put on the exam. So rostral midbrain, I will show you the caudal midbrain looks a little different, lots of similarities. I'm not going to ask you to identify the difference between like rostral, mid, and caudal pons. They are in your textbook. We're just going to have one section through the pons. But I am going to ask you to identify the difference between the rostral medulla and the caudal medulla, and that's because they look so different. Okay, the rostral medulla. What is the feature that is the most obvious, that the only place you see this structure is the rostral medulla? To me, this is called the inferior olive. From the outside of the brain, someone must have thought it looked like a little olive. So at any rate, the inferior olive, it's like this ribbon-like nucleus here, and it's the only place that we see it. Okay. Some people also think, I think that's the most obvious, some people also think there's like little eyes and like a little nose and a like mustache there. Can you see the little person there? Yeah, okay. So, but I think the inferior olives are the most obvious. Okay. And then when we get to the caudal medulla, looks totally different than any other level. So again, and I'm spending some time here because you are going to be responsible for then not only identifying the levels, but where the different pathways are. So you want to, like early on, when I put one of these images up, kind of have a sense of what you're looking at. The feature that jumps out, again, these are just, you might find something else that jumps out at you and use that, but the feature that jumps out at me in the caudal medulla are these three pairs of nuclei. And we're going to identify these three pairs of nuclei, you know, over the next few weeks. If you look at the other levels, no other level has three pairs of nuclei like this. So just one other point that I want to make before we move on from this slide is that by convention, what you see on the upper sort of part of the screen is dorsal. So this is the dorsal aspect, and what you see on the bottom of the screen for each section is ventral. Or we could say dorsal or posterior, right? Or ventral or anterior. So this is the same organization. It's not to say that you couldn't open a textbook and they have it turned around, but it's just like by convention, most of the images in your textbook, this is how they're oriented. This is how they'll be oriented on the exam, and you should know what's dorsal and what's ventral.
Dorsal fins.
And when you think about all of the spinal cord cross-sections that you have viewed, right, whether it was in neurohistology or neurophysiology, right, does this look familiar? Okay, good. This, right, this is dorsal. So again, it's the same organization. So this is dorsal, or the back or posterior, and this is ventral or the front or anterior. So really that's just to say you're used to looking at sections where what you draw on the top or what is shown on the top of the screen is the back, is dorsal, and what you see down here is ventral. Same thing in these. Dorsal, ventral, dorsal, ventral, dorsal, ventral, dorsal, ventral. Okay. Any questions? Yes?
Could you repeat up to what part of the brain the dorsal will be posterior versus dorsal will be superior?
Oh, okay. So the question is about where do we have the transition from dorsal being posterior to dorsal being superior above the midbrain. So once we get above the midbrain, then our terminology changes. So for the midbrain, dorsal is synonymous with posterior. Okay, so these images, these images are from accompanying photographic atlas that accompanies your neuroanatomy textbook. The other point I just want to make is that there is an electronic copy of the photographic atlas through the library. So if you just Google, if you're interested in like looking at some additional images, you can just Google, which I think is in your syllabus, just Google like the Nolte textbook, and there are two options. There are two different Nolte textbooks that come up that you can get free. I say free, but you're paying for it with your tuition, so it's really not free, but you can get it as a BU student. Okay, so we are not going to memorize every single thing on this slide, but I did want to give you some larger images of the cross sections. Eventually, it would be good to look at these, when we're talking about different pathways, seeing if you can find it, and then you can check yourself with this schematic here. Okay. So at any rate, here you can see just another image of the caudal medulla. Here's the rostral medulla. Here are those inferior olives that I mentioned. Here's the pons. Oh, and so here you can see the fourth ventricle, right? And then the cerebellum behind it. So here we're looking, all of this is associated with the cerebellum. Everything behind this is the cerebellum, right? The fourth ventricle and the cerebellum. And you get that sense here. Here's that caudal midbrain. I won't put these on the exam, but you can see that the caudal midbrain still have the cerebral aqueduct. It looks quite similar to the rostral midbrain that we did look at. Okay, how you doing? All right, so here we are looking at the brainstem, and we can see a couple of different levels, right? So here, this is the rostral medulla. This here happens to be the rostral pons. Again, not different level of the pons, but it doesn't matter so much, but you can see that very bulbous ventral portion, and then this is the rostral midbrain. You can see those inferior olives. So I want to talk a little bit about terminology that is associated with general regions of the brainstem. I am not going to ask you to, identify the specific boundaries of the tegmentum, but I do want to just generally talk about this region, the tegmentum. So what are we looking at in this particular image? We're looking at three general regions of the brainstem from a dorsal to ventral sort of trajectory. I'm going to start with the tectum because it's like the easiest to describe. The tectum is the part of the brainstem that's behind the ventricular system. So what you can see, I guess I just drew right through it, but here's the cerebral aqueduct. So this is the tectum. Okay, so then we have more anteriorly, right, or ventrally, we have what's called the basal portion. And when I think about what's shown in red, in this red region, we have circuitry associated with pathways that go to the cerebellum, and we have motor pathways. So everything that's anterior to these inferior olives is part of the basal brainstem. This bulbous ventral portion of the pons is part of the basal brainstem, and those cerebral peduncles, those little feet that stick out, that's part of the basal brainstem, which means everything that I haven't mentioned is in the tegmentum. Everything in green is in the tegmentum. I only bring up this green region, the tegmentum, because when we talk about pain and temperature, it's helpful to have a general sense of where this area is. Okay? So I think I'm just going to leave it at that. We're going to follow a pathway through the tegmentum, as I mentioned, and just have a general sense. We've got this region behind the ventricular system, the tectum. Then we've got this front region where we have a lot of motor pathways, and then we have the rest of the brainstem, which is the tegmentum. So it's a general region. Okay. So I just want you to understand that we have these general regions. Okay. Within the tegmentum, this general region, we have what's called the reticular formation. So when I think about the reticular formation, the way that it's often described, well, it's the core of the brainstem. So as we look at these images of the brainstem, so here you can see we're looking at two sections of the medulla, the caudal and the rostral medulla. We're looking at two sections through the pons, and then this is the caudal midbrain and the rostral midbrain. So we're just looking at two sections of each region of the brainstem. And this pink region, in through all of these images, this represents the reticular formation. We will, at certain times, talk about the reticular formation. And we'll think about the term reticular, right? Like when we've used the term reticular in histology, it's like a network. It's like this disorganized network, maybe a framework for cells, right? So the reticular formation got its name because it looked like a mess of cells and axons. And it's really become, at one point people thought, well, maybe it doesn't really do that much. We can't really figure it out. But really, what we now know about the reticular formation is that we know very little about the reticular formation, that it's such a complex neuronal network. And actually, I'm going to skip forward 2 slides because I want to show you, just to give you a sense of the complexity of the reticular formation. So again, what's on this slide is not like details to memorize. It's just to give you a sense of the complexity. So what we can see here, this is a single neuron. That red arrow is pointing to a single neuron in the reticular formation of a rat. Okay. So this is a single neuron in a rat, and it's in the reticular formation in the pons. And what do we see? We see that this neuron has axons that go all the way down to the spinal cord. So this is going down to the spinal cord. This is the anterior horn of the spinal cord. It goes to a nucleus in the medulla. So maybe some of you have heard, I think you talked about nucleus gracilis. Does that sound familiar? Okay, that's nucleus gracilis. You can see it also has these other projections, right? It also, right throughout the brainstem, it goes up to an area called the periaqueductal gray. It's passing to the hypothalamus, and it's passing to multiple thalamic nuclei. So when I look at this image, it gives me an appreciation for why early neuroscientists called it the reticular formation, right? It's just one neuron, right? So we've got millions of neurons in the reticular formation. This is just one in a rat. So it really gives, I think, an appreciation for the complexity of the reticular formation. And again, here in 2022, we really know very little about the reticular formation. We do know about some functions, and I will talk about it a bit as we go through the course. Okay. So we know that it modulates and integrates a lot of information. We don't always know how it does that, but many reflexes, different pathways pass through the reticular formation. But some of the general functions, it regulates visceral functions, so cardiovascular and respiratory functions. And to do that, it's going to have communication with structures like the hypothalamus and locations of autonomic neurons that we'll be talking about probably Tuesday, maybe a little bit today. Okay. And that's the lateral horn of the spinal cord. So when we talk about the lateral horn of the spine, actually, we will talk about the lateral horn of the spinal cord very shortly. But at any rate, there are autonomic neurons there. Okay, alertness, attention, concentration. So there are regions that project to a region called the prefrontal cortex, again, that we'll talk about later. But I do want to mention what can happen with damage to a portion of the reticular formation. So when I'm talking about like alertness, right, and really consciousness, our ability to be aware of stimuli, that's associated with this reticular activating system, which is part of the reticular formation. So a part within the reticular formation, we have what's called the reticular activating system. And this region of the brainstem projects out to the brain and mediates essentially consciousness. So when you think about somebody that has, that perhaps is in a coma, right, where they're not aware of stimuli, that can be associated with damage to the reticular activating system or damage to the pathway up to the brain that affects the cortical areas that are important for alertness. So we'll talk about those cortical areas, mainly prefrontal cortex, later on in the course. But so when you look at this region or this bullet here, it's modulating the cortex, right? So it projects to the cortex and modulates the cortex. And we will talk about, it does have an influence over motor function, and we'll specifically talk about the reticular formation in a couple of the nuclei and what we know about its coordination of motor output. Okay, so that's a little bit about the reticular formation, again, in the core of the brainstem. Okay. Any questions? Okay, why don't we take about a 7-minute break or so? So we'll take another break, and then we'll have, let's start at 22 minutes past, just to make that you can see the brain with the spinal cord here. And really the point, we'll start by looking at the gross anatomy of the spinal cord. You're comfortable. Neuroanatomy. I realized that when we talked about the organization of the spinal cord, that was about 3,000 hours of lecture ago for you all. So let's just kind of go through it, make sure you're comfortable with the content. Okay, so we'll start by looking at the gross anatomy of the spinal cord. This is just an image. You can see the brain with the spinal cord here, and really the point, just to make that emerging from the spinal cord, we have pairs of peripheral nerves. So those that emerge from the spinal cord itself, we call spinal nerves. Okay. And that's what we can see, this gross specimen of the spinal cord. What we can see are nerves emerging from the posterior aspect, the dorsal aspect, and then we also have nerves associated with the anterior aspect. And if you recall, does anyone recall, does the dorsal aspect, is it sensory information going in or motor information leaving?
Dorsal is sensory.
Exactly, good. Dorsal is sensory. So dorsal is sensory. So here we're looking at, then a cross-section, and we looked at this cross-section previously where we're looking
I have
at
no
the
idea.
dorsal aspect of the spinal cord. So the dorsal aspect of the spinal cord is receiving sensory information. Right? Here's the ventral aspect. And as I mentioned previously, when we were looking at the brainstem cross-sections, what's at the top of the screen, generally by convention, is what's dorsal or posterior. And then I just added an image here, just giving you a sense of the location of the spinal cord. In gross anatomy, we'll talk all about the vertebra and the vertebral canal, but what we're looking at in this particular image, you can see the sensory fibers that would be entering into the dorsal aspect of the spinal cord, and then here are the motor axons that would be leaving ventrally, and then sensory and motor fibers combine as they pass into the periphery. Okay? So that's what I have here, the spinal nerve. That's where we have the union of the motor and the sensory nerve. So most of the nerves that we see in the periphery are combined, meaning sensory and motor. So there's this point where we have to sort of talk a little bit about gross anatomy as we think about neuroanatomy. There are, when we think about the nerves that are leaving the spinal cord, we have 8 pairs of cervical nerves. So when we talk about cervical nerves, and I'll say, like, this is a cervical cross-section, just know we use the term cervical, it refers to neck, right? So it's somewhere sort of in the neck. Cervical fibers also go into the upper extremities. The thorax, these are the levels that are associated with essentially the ribs, right? Sort of the thoracic region, like the sort of where we have our ribs. Lumbar, this is the back. So I'm going to put here, I'm going to write neck and upper extremities. That's a terrible, let's cross that out. So neck. And then the upper limbs or the upper extremities. Again, this is very general. You're going to see I'm oversimplifying it when we get to neuroanatomy. Lumbar is the back, right? Sacral, we're coming down to the pelvis, but the lumbar and sacral regions also do the lower extremities. And so this only becomes important at times, but when we're talking about like a lumbar or sacral level, like many of you have heard of the sciatic nerve, maybe if you've heard of sciatica, people have pain down their leg. That sciatic nerve has contributions from some lumbar levels as well as sacral levels. So that's what I mean. Yeah, they're coming out of the back, but they supply the lower limbs. So at any rate, we have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal nerve segment. And if you've taken gross anatomy, you'll know the numbers don't exactly match up to the vertebral numbers. Again, you'll talk about that in gross anatomy. So I just put in a slide here in terms of thinking about the organization. So it sounds like you all remember this, that we have sensory information that passes into the dorsal horn of the spinal cord. And this is written out for you on the next slide. But this is the dorsal part of the spinal cord. This is where sensory information enters. And all of these sensory fibers, what kind of neuron did I just draw here? A pseudo-unipolar, exactly. This is a pseudo-unipolar neuron, right? There's some stimulus. It could be a pinprick. It could be light touch. Whatever it is, that information, right? There's an action potential that's traveled along this axon, right, and into the central nervous system, right? We have sort of this entire sort of fiber that I've drawn here can carry an action potential. And then this is the ventral horn. We have our anterior horn cells. We can call them alpha motor neurons, anterior horn cells. These are motor neurons that pass out. I don't know why this keeps skipping forward. Sorry about that. These are motor neurons that will pass out to skeletal muscle. Okay. And then together, the two of these form our spinal nerve. So the next slide just draws this out for you. And what we're looking at here, I showed this to you very early on, we're looking at the right upper extremity, we're looking at nerves, they're called, this is the brachial plexus, you'll learn it in gross anatomy what all those nerves are, but really the point is I want you to have a visual of, we're gonna say that there's some sensory stimulus, right? So maybe there's a bug touches your arm, right? It's going to be carried along one of these peripheral nerves, and that sensory information is going to go into the dorsal horn of the spinal cord. Maybe I want to pick up, a cell phone or a water bottle, right? That means that there's some signal that's going to come down from the brain, communicate with neurons in the spinal cord, right? Communicate with neurons in the spinal cord. This axon is going to head out. Well, this is the biceps muscle, and maybe I'm going to contract my biceps, okay? So just to kind of have that visual of it, I think can help as we're sort of learning the pathways. Okay, so the white matter and the gray matter regions of the spinal cord. Let's just take a look at those. So this region in through here, right, like the butterfly or the H region, right, this is our gray matter. And just as we drew it previously, right, we drew this and then we said, this isn't showing up as well as I'd like, right? If this is, right, an alpha motor neuron, it has to be in the gray matter, and the axon is passing out to the periphery. So at any rate, this is gray matter, this is a myelin stain, so everything that's in purple is where we have our axons that are either heading up, to the brain, up to the brainstem, for example, or up to the thalamus, or passing down to lower levels. So we'll be filling in the spinal cord over the next several weeks in terms of what kind of information is coursing through these different regions of the spinal cord. Yes.
In the brain, is the gray matter and the white matter reversed?
Ah, interesting. Yes. So the question was just in the brain, is the location of the gray matter and white matter reversed? So in the brain, the gray matter, the neurons are in the cortex, they're on the outside, and the white matter is on the inside. Here in the spinal cord, the gray matter is on the inside, and the myelinated axons are more on the outside. Yeah. Now, in the spinal cord, you might see a Nissl-stained section. This has to be a Nissl-stained section. Why does this have to be Nissl-stained? Because you now know that the center portion is where we have our gray matter, right? So most of the sections I'm going to show you are going to look like this. They're myelin-stained sections, but here you can see the myelin is purple. In this image, you can see that it's a Nissl-stained section. The neurons are purple. Okay. All right. Okay. So we do have different regions of the white matter and different regions of the gray matter. The various regions of the white matter are highlighted on the left-hand side of this slide. But we can at least see terminology that looks familiar to us, right? So this pink region is dorsal. The red region is lateral. So lateral, again, is toward the outside. Medial is like toward midline. So this is lateral. And then the blue region is ventral. Again, we could also say this is dorsal or posterior. What's in blue is ventral or anterior. The term that's new is funiculus. So this is just a general region. We have the dorsal funiculus, the lateral funiculus, and the ventral funiculus. The other term that is sometimes used is column, which I guess is a little bit, easier to say. But dorsal funiculus, your textbook very often uses funiculus, but it's just a region. I should look up that word origin. I'm not exactly sure what that word origin is, but at any rate, they're just general regions of the white matter. These funiculi are comprised of axons that travel long distances. Okay, so they might go up to the thalamus, or they might go up to a brainstem nucleus. And that's in contrast to these, what are called propriospinal fibers. So these propriospinal fibers begin and end in the spinal cord. These axons don't leave the spinal cord. And they think about, like, they're coordinating reflexes. So if I step on a tack, right, there has to be coordination of reflexes at multiple spinal cord levels. Because what happens when I step on a tack, right? I am going to pick up, exactly, if I pick up one foot, I better extend the other, I'm going to crumple to the ground, right? So there has to be coordination at the level of the spinal cord, and that's through those propriospinal fibers. So these stay in the spinal cord, what's in gray, and what's in these other colors, blue, red, and pink, they leave the spinal cord. So those are some regions of the gray matter. We also have specific regions of the white matter. There are 10 regions to the white matter. They are called the laminae of Rexid. We will learn some of them, not all of them. You are only responsible for them as we go through a specific pathway. What I want you to know now is that we have 10 laminae of Rexid. Okay? What you can see is that they are numbered from dorsal to ventral, and that we have, and then around the central canal of the spinal cord is number 10, but you can see there's an organization from dorsal to ventral for the most part. So the white matter regions, I want you to understand we have these general white matter regions, right? 4 of them I mentioned, and then we have these 10 laminae of Rexid. We will learn the names as we go through the course. And again, we're not going to talk about all the laminae, just some of them. Okay. The last thing I want to talk about the spinal cord is that, and bless you, is the various regions of the spinal cord that we have variations. So we've looked at the spinal cord. You looked at it in neurophysiology. We looked at it together in neurohistology lecture. There are some differences. So here you can see really from the top down, right? So this is when it's helpful to know cervical, thoracic, lumbar, and sacral. So we can see C3, C5, C8, so several cervical levels, a couple of thoracic levels, a couple of lumbar levels, and a sacral level. And probably what jumps out at you as we look at the white matter, well, what jumps out at you with the white matter? There's a lot more of it in the cervical levels, right? And it's hard, admittedly, we can't tell. We can't really tell how much white matter, that there's a big difference between maybe C5 and C8 or C3 and C5. But when we look at C5 versus L5, there's a huge difference. Right? And the reason we have these differences, and I'm not going to draw any synapses, but generally, if you have information, I'm just going to say sort of generally, coming in from the neck, and then we have more sensory information maybe coming in from the upper limbs, and then we have sensory information coming in from the trunk. All of these axons have to get up to the brain in order to be processed. So what that means is that if you have a spinal cord cross-section down lower, you just have fewer axons, right? By the time you get up to a cervical level, everything from below is coming up, right? It's sort of like if I had a bunch of outlets, all along this wall, right? If I had one computer here, right, I'd only have one cord, but down here I'd have all of the, actually I did that backwards. Down here I might have, down here I'd have, just one coming out, but by the time I got down here, I did do that backwards, I'd have all the cords, right, kind of all coming up towards this side of the room. So at any rate, for sensory information, we're just adding, we're piling axons on at every single spinal cord level. And then in terms of motor, right, so let's say I have all these, again, I'm not going to put any synapses in, but I've got all these motor neurons up in the brain, right? Some of them are going to leave here, and some will leave in different locations, right? So that, again, by the time I get down to these lower levels, I'm only left with a few. Up here, I've got a whole bunch of motor axons and a whole lot of sensory fibers, okay? So we see these differences in the white matter. Okay. In the gray matter, we also see some differences. So what do we see? Well, we see cervical enlargements and we see lumbar enlargements. The levels you're not responsible for, these correlate with things like the brachial plexus and like the sciatic nerve, right? Why? Why do we have these big enlargements in the cervical areas and in the lumbar regions? Because we've got to innervate our upper limbs and our lower limbs. We've got to pile in that anterior horn. We've got lots and lots of motor neurons because all those nerves I showed you going to the upper extremity, the cell bodies are in the cervical spinal cord levels, or most of them are in the cervical spinal cord levels. Lower extremities, lots of muscle, right? We've got to innervate those. What about if we look at T5? Teeny tiny. Look at that anterior horn. What's it innervating? A few intercostal muscles, right? So we have what are called cervical and lumbar enlargements. Again, they correspond with the neurons that have to go out to innervate the limbs. The last point I want to make about the differences is that in the thoracic and upper lumbar levels, we have what's called the lateral horn. I put it in gray text here. On Tuesday, when we talk about the autonomic nervous system, it will not be in gray text. And that's because this is where we have cell bodies of the sympathetic nervous system. So what you can see in cervical and upper thoracic levels, we see what's called the lateral horn. I'll show it to you on the next slide as well. But what you can see at a cervical level, there's no lateral horn, right? Can you see how there's this prominent region in the thoracic? Oh, wait a minute. Here it is. 2 slides. Here's that lateral horn. This is the location of cell bodies of the sympathetic nervous system. So we'll talk about that on Tuesday. I did put in a slide showing you those alpha motor neurons. I know you've talked a little bit about this in physiology, but here's this anterior horn or ventral horn. Within the anterior horn, we have these big motor neurons. These are alpha motor neurons. We also call them anterior horn cells or ventral horn cells, but these are the alpha motor neurons that you talked about. The axons of these alpha motor neurons project out to skeletal muscle. Okay, and so we're back to the beginning.
Oh,
we're back
thank
to this
the
organization,
Lord.
right? So I think this is a nice, again, just a nice organization. You can go through it. Again, thinking about central nervous system, brain and spinal cord, and all the different subdivisions
Oh my God.
of the central nervous system.
I'm
okay?
so
we sort of started off with the
tired.
brain, and we made our way down through the brainstem, and then the
Oh
spinal
my
cord.
God.
So does anyone have any questions?
I think I might take a 4-hour nap.
Okay. On Tuesday, we're going to start with the autonomic nervous system, which is another.
Yeah.
It's very
I think
short,
that's the
maybe
move.
half an hour or so. It's kind of.
I'm gonna take like a
our last
sweet
general
nap.
overview lecture, and then we get into the pathways which I think is the interesting part, right? Where we start now taking this foundation and building on it through the.
That's the
pathways.
move. That's the move.
Okay, so bear with me through this introductory material. I know that there's a lot of terminology.
2
Try to get
hours.
comfortable with rostral and caudal and some of the sections, because next week I'll just start putting up and say, like, this is the rostral midbrain, and it just is helpful if you're comfortable with it. All right.
All right. That was one. That was one lecture. 7 more to go. Here we go. All right. I'm I'm gonna die for a little bit, and then I'll probably be back again in a few hours. Wah wah wah. Bum ba da ba.